The next thing that we covered was the immune system response. So here we had this diagram like this, where we've got time along this axis, and along our y-axis we have the amount of secreted antibody, which we can also call immunoglobulins. And these are going to be secreted by B cells. And we're not going to just look at any old one of these. We're going to look at ones that are specific for our pathogen of interest. Oops. Okay. So in this diagram, we get exposure to a pathogen here at this time. So I'm going to do this one in red. So here's our pathogen A will be in red. So after your first exposure to the pathogen, it's at this time point, about 5 to 7 days later, you get a really good immune response to it. So, and you can measure this by looking at the immunoglobulins that are circulating in your body. So, for example, you can get, a COVID swab done, or you can get a blood draw. And you'll see a lot of these antibodies, the secreted kind. Now, what's happening here is that your T cells and your B cells that are specific to this antigen are getting activated. So again, it's going to be the ones that are specific to pathogen A. So we say that they're activated. And what does that mean? When we say that they're activated, that means that one, they're doing clonal expansion. So the specific T cells and B cells that recognized the epitope. So specific to pathogen A means they recognized an epitope on pathogen A. So all of the B cells and T cells that recognize that epitope are now going to start to clone themselves to make more and more T cells and B cells that also recognize that particular pathogen. And when they do clonal expansion, they're going to make effector cells, and they're also going to make memory cells. Okay. Some of these effector cells are going to be the plasma cells. And these plasma B cells are the ones that are actually secreting this antigen that we see in this reaction. So these are the ones that we're really testing here. But we know that the T cells are doing the exact same thing. Okay, so now you've cleared your pathogen, and you see that the amount of antibodies you have is starting to go down. That's because the pathogen is cleared. But what we don't see here is that we still have the memory cells. So at this point here, we still have our memory cells. Our other cells are starting to die off, or effector cells. But our memory cells are still around. Our memory cells aren't secreting tons and tons of these antibodies, so we can't measure them on a graph that looks like this, but they're still there. And these are going to be really handy if you get exposed to this pathogen again. So now we're going to get exposed a second time to pathogen A. And if we look at the immune response, now instead of this lag time here, the immune response will be almost immediate. So instead of waiting 5 to 7 days to get a good response, within 1 to 3 days, you'll get a really good response. Okay, so here's our really good response that happens the second time we have the exposure. Now, just to see whether this is just because our immune system in general is activated, or if this is a specific response, we can also look at another pathogen. So what if this is our first exposure to pathogen B? Well, let's look at antigens specific to pathogen B. And what we see is we see almost the exact same response that we got on pathogen A's first exposure. So these would be our immunoglobulins against pathogen B, and we can see that those don't exhibit this fast, heightened response. And that really tells us that this second response is totally specific to pathogen A, and it's mediated by these memory cells that are still floating around in your body even after the pathogen is cleared.